So now let's talk about the type of media I can use to cultivate bacteria in a laboratory. Again, culture medium represents an environment in which microbes can grow due to the nutrients we supply. So we select the amount of nitrogen, the amount of carbon, the amount of salt, all the things a bacteria would need to grow. And those concepts were discussed in a previous virtual lecture when we discussed about the chemical requirements for growth, and we also discussed the physical requirements for growth. Now there are three terms that are defined here, two of which we've discussed already. Let me quickly address those. We've defined inoculum at the beginning of this lecture. It's the introduction of microbes into medium. So I can take an inoculum from a nasal passage. I could take an inoculum from blood. I could take it from a prepared sample in the laboratory. When I grow the microbes, I grow them in what's called a culture. Microbes growing in the medium in which I introduce it represents a culture. That culture can be used to stain. That culture can be used for further growth analysis. Now, the word sterile is important. A sterile environment represents one in which there are no living microbes. It's very difficult to create sterile environments due to the ubiquitous nature of microbes all around us. So only certain things can be sterile. Prepackaged instruments are, for example, things that just come out of the autoclave, as we'll learn this semester. They are sterile. But it's very rare, especially in a clinical environment, that we're completely sterile in all cases. Certainly a surgical room, you would hope is sterile. If you're in a burn unit, you hope that's a sterile environment. If you're in a neonatal intensive care unit, you hope that's sterile. But it's very difficult to maintain that level of sterility, again, with the ubiquitous nature of microbes. So now let's talk about culture media. One important concept here, and we've discussed it earlier, is that we could have liquefied media, or we can also have solid media. Now, as we'll learn in lab, liquid media is great for fresh cultures. It also allows us to cultivate a large number of microbes. Now, solid media, however, has its own uses. We saw in streak plate isolation, solid media on a petri dish can be used to separate mixtures of bacteria. We also can use solid media to store cultures over long periods of time. Now, what's the difference between a liquid culture and a solid culture? Well, let me first introduce what they have in common. What they have in common is they're both growth media, which means they have all the nutrients, they have all the salt, everything a bacteria needs to grow. But the one difference you have between them is the presence of a polysaccharide called agar or agar. Agar is actually isolated from the cell wall of algae. It is added to a liquid media in order to solidify it. We often find solid media in petri plates. We can use them in media called slants and even use it to cultivate anaerobes using agar deeps. Now, the reason that we can be so successful using agar is agar cannot be broken down. That's what metabolized means here. Microbes lack the enzymes to break down agar. If they had the enzymes to break down agar, no longer would the media be solid, right? It would be liquefied. Every semester, I can't tell you how many students get this wrong on the exam when I ask about agar. It cannot be broken down. Hence, that's why it makes a good solidifying agent. Now, when we sterilize the media, it's going to be in a liquid state. So when you raise the temperature to 100 degrees Celsius, then as it cools to room temperature, it reaches that 40-degree threshold in which it solidifies. So let me show you a picture here of a slant. So what happened here in a slant, the way we prepared it is we autoclaved it. We raised the temperature of the media to 100 degrees. Therefore, it's in a liquid state. We poured it into the test tube, and then we allowed that test tube to sit at a 45-degree angle. And as it sat at the 45-degree angle and cooled, we can see it cooled and created this slanted appearance, hence the name. And it's this slant that's very useful in storing cultures long term. So now we've talked about the solidifying agent, but what's in here that makes this media? There are different types of media we're going to discuss in this chapter. Now first let me highlight the last three here: selective media, differential media, anaerobic media. There's already a virtual lecture on these three topics, so I'm not going to take too much time in this particular lecture to review those two, or excuse me, to review those three. But I'm going to focus on are these two types of media, defined and complex. So why don't we do this? Why don't we define again for you what media is? Why don't you define it in the box that's going to pop up on your screen? That's right, media has all the nutrients required for growth. It allows us to generate large number of bacteria in a short period of time. So now let's discuss the two major types of media. First, we have a chemically defined media. It gets its name because its chemical composition is known or defined. Here's a recipe for a defined media. Notice here, how much sugar do I have? Well, I have 5 grams. How much of this buffer do I have, the ammonium phosphate? I have 1 gram. How much salt do I have to control water balance? 5 grams. How much magnesium sulfate, another salt, do I have? 0.2 grams. And then I make that up into 1 liter of water. Now, if you look at the recipe, will this defined medium be a solid or a liquid medium? That's right, it's going to be liquid. Due to the absence of what substance? That's right, there's no agar in the recipe. Now, why would I use a chemically defined media? Chemically defined media is useful to cultivate a narrow range of bacteria. So in this example, this is a medium that's useful in growing E. coli. So if I take a sample that I know has many different bacteria in it, but I just want to grow the E. coli that's in there, I'm going to use a narrow range of nutrients in which E. coli grows best. Streptococcus, for example, which is another bacteria, would not be able to grow in this chemically defined media. So what that means is we can only grow a certain type of bacteria, which makes this useful. Now, a complex or an undefined media uses extracts. Now, an extract represents an organism which has been digested. The cells of that organism, as they're digested, release these cytoplasmic contents, and in the cytoplasm are vitamins, cofactors, carbohydrates, all of the nutrients that are stored in that cell. As we break it open now, they are released into the environment. So for example, what it means to have a yeast extract is that we've taken these yeast cells, we've lysed them open, and all of the growth factors that yeast cell has to support its growth is now released, and we can use it to cultivate our own bacteria. So for example here, we have 5 grams of partially digested protein, which will supply the amino acids. We have 8 grams of sodium chloride. We have 1 liter of water. We can see this is solid media. We have 15 grams of agar. And then notice here we have the presence of beef extract. Now here's where I think students have trouble, and even some instructors have trouble with this concept as well. I know exactly how much beef extract I'm adding. I weigh out 3 grams of that. So some of you may incorrectly say, well, I know all the ingredients. I know there's 3 grams of extract. This must be a defined medium. But if I were to ask you how much vitamin B is in that beef extract, could you tell me? Could you tell me how many grams of carbohydrate we have in that extract? Of course, the answer to those questions is no. An extract has what's needed for cells to grow, but we don't know exactly what it is, what the chemical content is. That's why it's complex or undefined. This, because of all of the cofactors, vitamins, everything that's in this extract, a lot wider range of bacteria and its growth is supported by undefined media. So if I take an organism out of the soil, let's say I just take a soil sample and I want to try to grow the bacteria there, I'm going to choose an undefined media because I don't know exactly what it needs. So we really only use a defined media for those bacteria that are well characterized and whose growth requirements are strictly known. Where an undefined media is good if I'm not sure exactly what's needed, I use extract to supply what's needed. Now this leads then into selective media, but let's just define what selective media does. Is it inhibits the growth of unwanted organisms while supporting the growth of wanted organisms. Now generally for selective media, we generally can use defined media, which only supports a narrow range of growth, or we can use some agents. For example, you can use lots of salt because we know most bacteria can't grow in high salt. Or you can use acidic pH because we know most microbes can't grow at acidic pH. That's what we've done here in this plate. Here I have a soil sample with a pH of 7. Notice all the bacteria growing in that soil sample at that pH. But take that same sample and add it to this selective media that has a pH of about 5, and notice how much less growth we have total here. And notice what's growing looks very different than at pH of 5 than what was growing at a pH of 7. See, microbes called fungi can grow at acidic pHs. Here I can't see the fungi. The bacteria are overwhelming the sample. But now what I've done is I've suppressed the growth of the bacteria, so now only the fungi that were in my sample can grow. That's a great use of selective media. And now let's say I wanted to define a new fungi. Maybe I want to find a new antibiotic growing in the molds here. Now I could characterize that mold and search for that antibiotic. Whereas if I just grew it at 7, I can't find the molds because they're being suppressed by the sheer number of bacteria that are present. Let me just introduce the concept of differential media. What happens when more than one type of organism can grow? How can I identify them? Well, I can differentiate them using this special type of media. Here's a great example. I have 3 different streptococci organisms on here. I have A, B, and C. Now they all grew, so this plate supports growth of strep. These 3 types of strep look very different from one another on this blood agar plate. Therefore, I can differentiate among the different bacteria by simply characterizing their growth. I know A is going to be Streptococcus pyogenes. You're going to learn that in lab as well. I know B is probably something like Strep sanguis. I know C is something like Enterococcus faecalis. Now, Professor Weber, how do you know that those 3 bacteria are represented by A, B, and C? Because I know what Strep pyogenes looks like on this plate. It breaks down all the blood. I know what Strep sanguis looks like on this plate. It only partially breaks down the blood. And I know Enterococcus looks like on the plate. Why? Because Enterococcus can't break down the blood. So I can distinguish amongst similar bacteria based on their appearance on differential media. Here's another great example. Their ability to break down sugars. Here I can see 2 closely related bacteria. These are both gram-negative. And they're both rods. So if I were just to do a gram stain, let's say I have 2 separate samples. I have sample A, sample B. And I did a gram stain. What color would both organisms be in a gram stain? That's right, they'd both be pink. So I can't just stain and say, oh yeah, there's alkalogenes, there are E. coli. They're both going to be rod shape and they're both going to be pink. But now if I put them in a test tube with carbohydrates, notice their ability to break it down is going to impact what the media looks like. E. coli can break down. It's positive for the breakdown of that carb. Alkalogens is negative for that. So now I could distinguish amongst similar bacteria based on the differential media which I've used here, specifically carbohydrate utilization. Now, as we go on in the virtual lecture I've given, I talk about blood agar, I talk about mannitol salt agar, I talk about EMB. For our case studies for Chapter 6 homework, case study for Chapter 5 homework, and on the exam, make sure you view the virtual lecture and know how MSA works, know how EMB works, and know how McConkey agar works. Because these types of selective and differential media help us to identify clinical specimens by name. And so in a case study, I'm going to use selective and differential media in order to figure out what's causing a food poisoning outbreak, or what's causing an outbreak, a rash outbreak due to a hot tub that's contaminated. I mean, I can set up scenarios in which I identify organisms using selective and differential. So I really strongly suggest you go listen to that virtual lecture.